Swadika. Good morning, ladies and gentlemen. My name is Pajaree Chariyavilaskul. I'm a medical doctor and a clinical pharmacologist. I'm not an immunologist or any person who are experienced in HLA, but part of my work is related to HLA. That's why I'm here. So I'm trying my best to present the clinical implementation work regarding to HLA long-read sequencing in Chulalongkorn Hospital. First and foremost, I have no conflict of interest, non-financial or financial, to disclose. So I would like to divide my talk into 3 sessions. First one is the importance of HLA long-read sequencing, and then we'll move on to the database that we have in Chulalongkorn University, and give you some example of the work that is already implemented and ongoing at the moment. So you already heard, well, it's the HLA conference and the HLA day. So all of you know the HLA is a very complex genetics regions in human genome, and it has its role in a lot of disease and conditions like transplantation, disease susceptibility, and also some personalized medicine. So challenges for accurate HLA genotyping is a key for clinical application. I've reviewed the methods of trying to genotype for HLA. From the past, as you can see here, that we have serology before I was born. And, well, yeah, 10 years before I was born. And then we move on to the intermediate resolution, something like Luminex, which is quite easy and widely used in our country. And then we move on to the next generation sequencing, which is highly important these days. And the second generation sequencing will give you high resolution HLA genotyping, for example, the Illumina or Ion Torrent. And then we have third generation sequencing, for example, PacBio or Nanopore will give you the ultra high resolution. The thing is, our group is very interested in the ultra high HLA resolution because we think we are doing something about personalized medicine, so we think that, well, if we can read the gene by the biggest step, that would be perfect for everyone. So we decided our studies trying to sequence the 8-digit HLA typing for every single person entering into our cohort. You may have heard that Thailand has the campaign called Genomics Thailand. Phase 1 is already finished, and we are going to start for Phase 2. Genomics Thailand platform, they use a short read sequencing, and our platform would be a lot more detailed than the genomic platform one. And also, HLA long read sequencing is a method of the year a few years back, and because it gives more information, it improves haplotype phasing, resolving both maternal and parental HLA alleles with without any phasing error by the computational things. And it detects a lot of structural variation and relatively fast turnaround time. It's not that really fast, but it's faster now. To me, the application of HLA long-read sequencing can be implemented in transplantation medicine and pharmacogenomics and drug response, disease-related like infection or autoimmune disease, and cancer immunotherapy, and also some population and evolutionary genetics. So far, here in Chulalongkorn Hospital, we have implemented several studies related to HLA long read. We have cohort of transplantation, heart and kidney, and also we got some samples from donor registry patients. And we finished the study with red cell aplasia, which I will show you later. And also we are continuing doing some hematologic malignancy cohort, and also TB, which is highly prevalent in Thailand. The ongoing studies in SCAR is also ongoing, and also we have some patients, like 100 of them, having long-read whole genome sequencing, not just HLA sequencing. And so we identify, comparing to intermediate resolution, we can identify better HLA alleles from HLA class 1 and 2. For example, for HLA-A, you can see on the left-hand side, your left or my left, your left. For intermediate resolution, you see that HLA-A 1101 would be the most HLA-A found in Thai cohort. And this figure holds true until you get to high resolution, but you see the HLA 0101 would be the highest amount of alleles found in Thai cohort, and it's split to A01, 01, 07 that contribute to the cohort as well. The same figure, you can see it from HLA-B like this. We have a lot of HLA-B 5801, which is related to allopurinol scar, the skin reaction related to allopurinol. And you normally see in literature that Thai patients will have about 5 to 10% of HLA-B 5801. But then when we look into the high resolution, we know that it's actually HLA 5801 that contribute to the severe cutaneous adverse drug reaction in Thai patients. I'll show you the data later. And also the same figure for DQB1 and DPB1. This is just a general idea. For clinical implementation, as I'm a clinical pharmacologist, so I'll focus on the pharmacogenomics and the response. The first example is pure cell aplasia, which is the conditions where the patient has just only normochromic, normocytic anemia, happen after having erythropoietin use. Pure cell aplasia can be from congenital or primarily acquired, but drug and toxins can cause a secondary acquired pure cell aplasia as well. And several drugs involved, especially recombinant erythropoietin. In Thailand, we have millions of people with CKD, and 50% of them would be CKD stage 4 or 5, which require the use of EPO. And we have PRCA cases, roughly around 2,000 to 3,000 cases per year, which is quite a lot compared to the incidence in the United States. So this is a big problem in Thai healthcare for CKD patients, because the pure cell aplasia patients would require packed red cell transfusion every 1 or 2 weeks for continuously, and no spontaneous recovery, and a lot of them die because of this condition. So we gather 157 cases with 56 control to see whether the HLA has its role in predicting or associated with this condition or not. There are some other factors that might relate to PRCA, and we already tease out all those things, like the type of EPO used in our country, or the way we inject the EPO into the patients, and also the exposure time. And we found from the long-read sequencing that HLA-B 46010101 and DRB10901, 0201 are highly associated with the PRCA condition in Thai patients. And these alleles goes together in Thai patients. We did some haplotypes analysis as well. We also did the in silico molecular docking to see whether these alleles are really interact with the HLA and EB protein complex are interacting together, and we found positive results like this. Compared to other studies in the world, not many. I can find only 3 studies in the world because it's rare condition compared to other conditions in CKD. And in Europe and Canada, they found that the RB109 are associated with this condition in Chinese, and we've recently reported our study, and previously we report a small cohort of patients. That's why we put HLA into our attention, that we have DRB1 as a role for association. So these are the example of clinical implementation in PRCA. The next example is the severe cutaneous adverse drug reaction. I think all of you have heard that if you want to take allopurinol or carbamazepine, you might want to have your gene test for HLA-B 5801 or 1502, right? And it's already recommended by a lot of the regulatory in different countries. In Thailand, we are prevalent for these risk genes. For Thai patients, they already have tested for 1502, 5801, and 5701 if they are going to use these drugs. But again, we have a problem that some people, even they have all these genes, they don't have any complication, and we want to know why. Partly, they might have some specific HLA genotype that's specifically related to SCAR rather than just 5801. So we look into our database, and we found that, for example, allopurinol risk gene is 5801 for intermediate resolution. In our cohort, we found 3 genotypes, HLA genotypes like 01, 0101, 0103, and 0110, and with different allele frequency. And after we analyze for odds ratio, we see that the only patients who have 5801, 01, 0101 will be risk for having severe cutaneous adverse drug reaction when using allopurinol. Those 2 groups can use allopurinol safely. This is the new thing that we are trying to implement, and the cohort is we continue gathering some other cases to make our cohort bigger to confirm all these things. So those are the examples of the work currently ongoing in our hospital. There are some other challenges for HLA long read that I would like to mention here. First, the higher error rate for doing HLA long read sequencing, but the recent improvement in base calling and error correction are underway, so it might resolve this issue. And the cost consideration is relatively high compared to the short read sequencing and some other intermediate resolution HLA genotype, so we need to find a way to reduce the cost, and also the standardization and regulatory approval for reimbursing all these HLA long read. So in summary, long read sequencing represents a very promising transforming approach for HLA typing in clinical application, offering very good in-depth accuracy, phasing, and variant resolution for HLA. However, the further validation and cost reduction still required for supporting the use in clinical practice. And I would like to end my talk here showing the team, Dulan team, who are conducting all the work with me. And I would like to thank all the audience and happy for comments and questions. Thank you.
Thank you, Professor Pajaree. And now it's time for questions.
It was a really interesting talk. I was wondering if in the future you would imagine that you would start, say, allopurinol sensitivity testing at a low resolution first, and then get that 5801 result, and then test it at high res, or if you'd prefer to do long testing up front for everyone and not have to do it twice for some patients. What do you think would be the better way to handle that?
For now, it would be the low resolution first because of the cost. But then I think in the next few years, we might be able to do the long read sequencing with an affordable price. So that would be perfect for everyone.
Thank you.
Just I have a comment regarding your last statement, long read sequencing being less accurate than short read sequencing. That depends on which chemistry you're using. They are not the same PacBio versus Oxford Nano, the accuracy is different, number one. And the chemistry used within PacBio is also different. With the current hi-fi algorithm with PacBio, we do in parallel, we get similar levels. Sometimes we have to rely on PacBio sequencing for difficult regions.
Thank you.
So as technology evolves, especially the new technology of the new chemistry of PacBio that's coming in 2 months, that will be even better. The limitation we had with long read sequencing was the sample quality and DNA quantity. So now with short read sequencing, you could get a sequencing result with lower concentration of DNA. However, PacBio's new chemistry is also addressing this issue. So this field is keep evolving.
Thank you. Just to let you know that every single data here is from PacBio. But I'm not a PacBio person, so I have no conflict of interest.
Thank you for a question from the audience and from our speaker, Professor Pajaree.
ขอบคุณค่ะ
Let's have a big round for her.